This is Making a Scientist, the podcast by young scientists, for young scientists, and hosted by me, Alex Ainscough. This week, my guest is Dr Mick Jones, who is a reader in molecular medicine at Imperial College London. Mick has spent his career working in genome analysis, and he was one of the early researchers in DNA sequencing, cloning, and protein chemistry. Mick's career has been fascinating. He had the opportunity to work in two separate groups where the group leaders subsequently went on to win Nobel Prizes. However, he stayed true to himself and followed the work that he was really passionate about, and he later ended up rubbing shoulders with many other Nobel Prize winning scientists, including Fred Sanger and Aaron Klug. This is one of the most deeply honest and thorough interviews from one of the most brilliant and humble researchers that I know. Mick gives a whole load of incredible advice and discusses the various decisions he made during his career, including about when he moved to Denmark for a couple of years. All of that money that he spent on Danish lessons, and he still can't speak the language. He tells the story much better though, and I'm really looking forward to sharing this episode with you all. So let's begin.
Not at all. Pleased to do it.
So what we'd like to start with is if you could, well, I know you're a semi-retired man now, but if you wouldn't mind explaining what your research was and how you've made an impact in the world.
Okay, I guess my area of sort of research and interest I would put down as genome analysis. In other words, applying DNA sequence analysis to understanding gene function. And I've dabbled in lots of areas. I use my expertise in DNA sequencing and allied cloning, oligonucleotide synthesis to sort of establish that expertise at Hammersmith. And if you go back to the sort of the 1980s, you couldn't buy oligonucleotides. DNA sequencing was very expertise driven, labs would spend a lot of time doing it. It wasn't that everybody would do a DNA sequencing.
Sorry, when did DNA sequencing actually, when was that started to be a thing? When did it start?
I guess Sanger's paper was in 1977, so I guess the sort of into the early 80s groups started doing it, but it was still very labour intensive, using radioactivity. You were running gels like this, the autorads, and it was very time consuming. Not everybody wanted to do it, and not everybody was necessarily sort of good at it, but it was rapidly coming on. And you've got to remember, when DNA sequencing started, PCR had not been invented. There was a time when people did a lot of work before PCR was invented. And just to give you some examples, when I started doing my molecular biology in my first postdoc, we didn't know about intron-exon structure of genes. Very few genes had actually been sequenced. They were mainly simple viruses and a few bacterial genes. And, that recombinant DNA technology was only being able to clone genes was only just starting.
So just for our listeners, what's the difference between an intron and an exon?
Well, an exon is an express sequence in the mRNA, and an intron is an intervening sequence which splits up the gene. So when a gene is expressed, you make the whole of the gene with its exons and introns, and then you make an RNA copy, and then you remove the intron sequences and join the exon sequences together to give the message, which is then translated into your protein. Okay,
cool. So what's the point in an intron? Just to follow that up.
It's difficult. I mean, say one of the things about it allows you effectively to mix genes easier, to duplicate genes, to make variants. You can actually join sort of structural elements of a protein and to create new proteins by mixing exons together. So it is sort of nobody really knows necessarily why, but they're there if you want to go back through evolution. They allow things to be, I think, your cells to develop and you to be a more multicellular organism rather than being like a single cell bacteria. That's it. You're actually multicellular. You become animals, effectively plants. Become, organs with different heart, lung, all those sorts of tissues.
Yeah, definitely, and I suppose the also the way that the introns are spliced, like you can get a greater complexity of the protein. So one protein can have a whole bunch of isoforms, right? So it can take on many forms. Yeah. But just going back then to your oligonucleotide work. So I mean, to me, it's crazy because I just I log on to the Sigma website, I put in my sequence, I order it, and Sigma are really good. They pretty much come next day with my primers. So how did your efforts sort of translate into me being able to do this so quickly?
Well, I guess like a lot of other groups around the world, I mean, say at Hammersmith, when I started there, we applied and got some money from the Wilson Foundation, and we set up the Wilson Molecular Biology Unit. So from about the late 80s till the early 2000s, I ran a facility which would make oligonucleotides for anybody who wanted them. We charged more or less just cost of reagents to make them. And we started offering a DNA sequencing service. So people would send us their PCR fragments, their clones, and we would sequence them and again and charge them for that. So it was available for anybody on the Hammersmith campus. Obviously now in the 2000s, we could not compete with the price reductions and the capacity that the pharmaceutical, the industry companies now do. So, we basically we closed them down. But in the 80s, 90s, that was how we got research done at Hammersmith. So anybody at Hammersmith could send in a request and we'd make an oligonucleotide for them.
So what would these oligonucleotides typically be used for? Would they be for PCR? For would you then run PCRs for these people? Or?
No. We just make the oligonucleotides, and they would then use them. Doing the PCR effectively is quite easy, but you need to know the sequence to design your primers, your PCR primers. So we would make them, and I think a lot of work that went on at Hammersmith at that time. If it wasn't for us, they would have been delayed in not being able to get primers. We could make primers within 24 hours. Although we could only make 12 in any 24-hour period, but we had a technician who would just make them. That was all she was doing. And we had a technician who was running the early sequencing machines 24/7 effectively. So we could provide this service to people. And it just became, not cost effective to continue running it. We didn't have the capacity.
So, but still, to have this as a service, people clearly figured that there was such a demand for wanting to know the sequences of genes and wanting to do PCRs to find out what's the cell is a black box. I mean, it still is by all respect. Yeah. You don't know what's going on inside. A PCR only ever provides a kind of snapshot in time as to what has happened at a particular moment, but the sort of this technology that was developed and sort of was shown to have huge significance by your work in Hammersmith, that must have led to commercialization in some respect.
I guess a lot of it was again getting the fundamental understanding of genes. I think that's the whole aspect of sort of the molecular biology, and doing the sequencing and what PCR allowed you to do. Was to better get on with that. So I think my expertise in that area helped a lot of scientists, start and being able to make good research, and I was on quite a lot of papers, where we provided that expertise. That was, I think, was quite, a great scientific discovery. I think one of the ones that I'm really quite proud of was working with Dave Holden on developing his signature tag mutagenesis technology, which allows you to look at any pathogenicity genes in bacteria. And you can do a screen of all the pathogenicity genes in a bacteria using this signature tagged mutagenesis method that he developed. And I provided the tag technology that was used in that to allow you to actually help identify the genes that were causing the pathogenicities. That was, and we got a science paper out of that, which was quite nice. Publications in Science.
Yeah, that's awesome. Congratulations. Just as a quick follow-up then, that the tag technology, was that then applied to other fields?
You could, in more or less any bacteria, you could apply that technology. I mean, so initially it was done on sort of strep, but you could apply to any sort of microorganism. for doing that. So you could look at pathogenicity genes in any type of any assay. Because one of the problems is that when you're trying to find a pathogenicity gene, usually what you tend to do is you tend to knock that gene out. So you don't have the biology of that gene. But then the question is, well, you've knocked that gene out. How do you actually find which gene you've knocked out? And the STM technology allowed you to identify what gene you'd actually knocked out. And then you can go back and then study its function.
So then a potentially stupid question, because I know it's a problem that hasn't been solved yet, but could that be applied towards combating antibiotic resistance?
Oh yes, yeah, I'm sure you could use that. I think it's slightly changed because of the ability now to sequence whole bacterial genomes, literally in probably a week, less than a week. You can probably get a complete genome sequence of a bacteria, and then you can then use it. And I think it's the what has happened is it's just the incredible explosion in getting sequence data. Remember, when we were starting up to about in the 1980s, 1990s, very few genes had been sequenced. So any gene that you sequence was really an interesting, a new find and discovery. But now it's becoming sort of routine.
Which gene? Oh, sorry.
I was just going to say that actually, if you're looking at what this technology has now done now, you think now what is happening with the analysing the COVID viral strains. That is phenomenal, and it's because of building up this ability for PCR, automated DNA sequencing, that has led on to that sort of technology now being applied. In the 1980s, it would have taken you 6 months to sequence one viral strain of the COVID virus, whereas now you get them in a day.
But this is, going to get slightly sidetracked here, but this is one thing that I suppose the general public don't quite understand. And also, like us as scientists, we sort of have an underlying appreciation that many decades of research have gone into all of these different techniques and the evolution and the development of all of these techniques, such that you can just do sequencing, DNA sequencing overnight, RNA sequencing overnight, and or in a few hours, really, and then you just have the information there. Whereas, like back in the 80s, this is something that would have taken a long time. So the development of a lot of these vaccines, the development of a lot of personalized treatments that are in the pipeline, people don't trust them because they think, oh gosh, it's happened too fast. We've never been able to make vaccines before. I mean, well, yes, obviously, but we think about phone technology since the 90s and how much that's evolved. Like, why do you think medical technology hasn't? Anyway, that's just a side little rant from me. But I was just wondering, of the genes that which genes did you discover? Any particular genes?
Not particularly. I mean, I've worked on Epstein-Barr virus and one of the new herpes viruses, HHV6, and we just basically rattled off the boring genes. It's the glycoprotein B gene, the DNA polymerase gene, some of the more fundamental genes. But basically, I think it's genes interact with other genes, or the proteins interact with the proteins. So you need to get everything to really get a full picture of what goes on in an organism. It's not just sort of that, this is a really interesting gene.
Very true.
But actually, again, going back to one of the things I did early on in my first postdoc was actually work doing protein sequencing. And I was part of a group, and one of the main sort of workers, and we sequenced elongation factor TU from E. coli. So this is a protein which binds tRNAs to the ribosome for protein synthesis. And the group I was working with, they'd actually got the crystal structure, but they needed the amino acid sequence to get the full detailed structure. And that was really a fantastic piece of work and a really interesting discovery. But one of the things which we didn't appreciate is that protein is a GTPase. And because of looking at structure of other GTPase, you could actually model the Ras oncogene protein with the EFTU. So doing that fundamental on a bacterial protein has helped actually some of the work analysing oncogenes.
So a lot of this sounds incredibly impressive, but would you mind just breaking down what GTPases are, what their important function is, the oncogenes?
Well, basically, it's the GTPase is basically our proteins that use GTP in their in the biological reactions. So with EFTU, that binds a tRNA molecule, and then it binds that tRNA molecule onto the ribosome. And once it's got it correctly positioned, the way you release the EFTU protein from the ribosome is that it hydrolyzes GTP. And so it's like, I guess, a sort of an energy source. And similarly with ATPases, they use ATP as an energy source. GTP is just another sort of variant of that. And the link to the oncogenes is that one of the viral oncogenes that was discovered, I guess, in the 90s, is the RAS, which is a Rous sarcoma virus oncogene protein. And when you look at its structure, you can sort of mimic some of its structure with how GTP is bound to EFTU. So knowing that, you could realize that the RAS oncogene was also a GTPase. Okay. So it helped you understand that function of that oncogene.
Oh.
So that was quite interesting. That was sort of I missed out on all that sort of thing because I'd sort of left the group by then. But it was interesting to see that some discoveries in a certain field actually do impact and are useful in another field.
Totally, it's so interdisciplinary science, and I think this is what young people, if you're not aware of it already, that science is not just you have tunnel vision, you focus in on biology, you focus in on this, you focus in on chemistry, and you stay in this field. The real discoveries are at the interdisciplinary, in the interdisciplinary research. For instance, the Nobel Prizes in chemistry, the latest one was awarded for CRISPR. And that's all about gene editing. So it's a very biological thing. So yeah, it's just I think people need to get a really good appreciation of this interdisciplinary work. So you were an early adopter of this.
Yeah, I think you've just got to you've got to think the big picture rather than thinking too much narrow channel down. This is my research area, and I'm just going to concentrate that there. And I got that mainly because by providing the oligonucleotide and sequencing service, I got to know what lots of other people were doing. And, so that you sort of think of the big picture rather than just being concentrated on working on your protein, which you do for the next 20, 25 years, You've got to start thinking about that. And there are still some scientists who probably do continue working on a particular small portion rather than trying to think big. But you need you need both.
Totally agreed. So let's move on to the next topic. I'd like to just discuss about when you were young. So if you could start by please telling our listeners, about So where did you grow up? Where are you from?
Okay, I'm from Leeds, and I've not told that many people this story, but before I was born, my mother had my elder sister, and she was very ill. And she was told if she got pregnant again and went through with the pregnancy, she would probably die. She got pregnant again, and the doctors persuaded her to go have an abortion. And while she was being sort of taken into the operating theatre to have the abortion, she decided no, she didn't want to have the abortion, which I'm quite pleased about, because that was me. So I was not really, so at one point you could say I was not really, had the possibility of not actually being born at all. So I was quite pleased about that. So my mum was very ill afterwards. Okay? She's passed away now, but yeah, no, she was okay and brought me up. And so I started out in Leeds and went to a grammar school, did well at the A-levels, and then went on to university. And I think one thing I think you've got to bear in mind is that I went to university in 1971. And at that time, definitely less than 10% of 18-year-old pupils, kids, went to university. It's only a very, very small amount of people went to university in those days. Universities were much smaller.
So what did most people? What did? Sorry, just because I'm not sure. Then what did the other 90% do?
Well, they would have left with O levels. They would have got jobs. They would have just gone and got jobs.
But what was like the main sort of thing back then? Like, was everybody going into apprenticeships? Or
some would go into apprenticeships. Others, would get a job in the family firm or whatever. But basically, people were looking for jobs and not necessarily thinking of careers, or at least definitely not looking at further education in those days. There, I mean, so it has now mushroomed, which is great, but at that time there was much less students around as such, and universities were probably much smaller. Our chemistry year, we had about 35 students.
Is this in high school?
No, this was when I first year at university.
So just before you get to university, then, if there was such a small percentage of people who wanted to go on to university, then why were you in the 10%?
No, I guess I just liked science, and I was probably good at science. And in the sixth form at our grammar school, there were 20 students in the sixth form in the science year, and about 35 doing arts. So that was the total for the whole school, and probably out of that, only a quarter went on with those A levels to university.
Oh, right, okay. So did you have a careers advisor, or was it a family member who encouraged you, or did it just come from you?
I just like doing science and chemistry particularly, and so it was a case of when you applied, looking at the universities that were ranked that did good chemistry degrees, and I was just fortuitous that I chose them, and at the end of getting my A-level grades and what I'd been offered, I ended up going to Birmingham University to do straightforward chemistry.
It was just straight chemistry.
Just straight chemistry. Yeah, there was no option. There was a chemistry degree, and that was it. And again, I say there was only about 35 in our year. In that year, so there wasn't the you weren't doing like chemistry with biology, or chemistry with physics, or chemistry with business studies. It was just one degree chemistry. That was it. There wasn't quite this broad spectrum that you get now, which is a shame, because I'm not sure if I was applying now whether I probably wouldn't have done necessarily just pure chemistry. I may have actually chosen something else. Don't know what, but probably something else. But in those days, you're going to do science, then you either did biology, chemistry, physics.
Sure.
Biochemistry was still quite a sort of a new degree for people to be doing, and such like that. Never thought about engineering. The school never sort of mentioned me in the sixth form that why don't you become an engineer, or even doing medicine.
Okay.
So that sort of, I was just good at science and I enjoyed it. That's what I did.
Do you think you could have been pushed towards a more engineering or medical type degree if someone had suggested it to you?
I think probably. I mean, so I think when you're in the sixth form, when you're thinking about what to do, I mean, say it's usually the teacher that you feel has got most rapport with you, the one that you feel is you can talk to, who praises you, who pushes you. That's the one. And ours were the chemistry teachers.
See, that's the thing. It's incredible. You don't realise it at the time just how influential your high school teachers are on you. Like the ones that. Yeah, like you say, the ones that just have this nice rapport with you, then you then think, okay, I can, I'm doing all right at this subject. Maybe I can go on to study it. So at university. So yeah, it's crazy. But let's go back to your degree. So I'm assuming then your degree went well, and
yes.
Yes.
Yes. I mean, I got a first, and I was top of the year.
Oh, very well then.
So yes. Yeah, no. So fine. It was something which I found not necessarily easy, because you had to work at it, but I enjoyed it. I enjoyed doing the chemistry, particularly the labs, and I would say that's been all the way through my sort of career. Is being able to get into the lab and do things. Just so much fun.
It's incredibly important. That's if we've not even got onto our advice section yet, but I think it's too important not to acknowledge that the enjoyment factor in what you do, taking pride in your work. It doesn't matter what it is. It's just a general life lesson. But taking pride in your work and really, really enjoying it. That's the first step to doing well, to succeeding. So
yes.
Definitely. So you finished first. You finished first in your degree. What did you then consider doing? What did you actually do after you were graduating, or around about the time you were graduating?
Again, it was the chemistry departments at that time seemed to be taking the view that if we've trained this undergraduate student to give a good BSc, we want to keep them for PhDs. So I was approached by the head of department and basically said I could have a Science Research Council PhD scholarship, and I could choose whichever group I wanted to go work in, which was just really nice. So actually the group that I chose was the nucleotide group, the nucleic acid group, because they seemed to me the most sort of exciting and interesting chemistry that I'd come across.
So this was at Birmingham still?
This was at Birmingham, so I stayed and did a PhD in the nucleotide group there.
So did you have any other offers, or did you look at anywhere else?
I mean, I looked at sort of carbohydrate chemistry, but that didn't seem to have quite the sort of the appeal. I mean, and again, you remember in the 1970s, DNA, that side of things was becoming more and more sort of important. We were hearing about people doing the first sort of gene cloning experiments. DNA sequencing, was sort of, was there. We were hearing about the earlier methods that Sanger was working with, and the nucleotide group I was in, they were actually working out the sequence of the tRNA molecules as well. That was one of their sidelines, as well as nucleic acid chemistry. So that sort of got me interested into sort of what would become effectively molecular biology. That field.
So were they a big group? You've obviously dropped Fred Sanger's name in a couple of times. He was double Nobel Prize winner. I think he was at Cambridge at that time, but correct me if I'm wrong.
Not yet.
So he was. So did you ever consider going to Cambridge? Was Birmingham a big group at the time? Like this? It
was quite well. In some perspective, it was quite a large group. We had over the 3 years, there were sort of 9 PhD students. We had about 3 each year, and there was a postdoc and a technician. So it was quite a large group, and I think that was quite nice to have other people like you were working with, your colleagues, which is really important, that you getting on with your colleagues and having people that you can chat to and discuss your ideas. No, I hadn't really thought about going elsewhere, but again, because the chemistry department was basically, they were saying, come and work with us, we can give you the money and such like. Whereas I know other departments, they more or less are quite keen that their undergraduates, when they finish, they go out elsewhere and spread the word how good the Birmingham University is in their particular sort of degree. But no, I hadn't really thought about going away from Birmingham at the time.
Okay, fair enough. So then, you did your PhD. You graduated from Birmingham. But then, when maybe just if you could take us back to the mindset that you were in after you were about to graduate, or around about that time.
What, for the PhD?
For the PhD. So did you always picture yourself going down an academic track, or again did you then consider other career paths?
I suppose I thought at the time, as I'd seemed to have done really quite well as in my undergraduate degree, I thought maybe yes, an academic path was a way that I would go, but I hadn't sort of got any plans about I would do this topic and move on to do this, and I would eventually end up at a university somewhere. It was all just basically taking the next sort of 2 to 3 years at a time and seeing where they would lead. So I never had any particular defined plans. It was basically what was offered to me at the time, and to make a choice out of what was available, and then moving on from there and seeing what happened at the next stage. So I don't think I ever had any particular plans to say, in 20 years' time, I'm going to be a lecturer at Imperial College. It came there through the contacts and what was available at the time.
Fair enough. Okay. So then, you graduated from your PhD. What did you pursue after your PhD, and where did you go?
Okay, so because I've been working on sort of nucleic acid sort of chemistry, I initially, when I talked with my supervisor about going next, is that one of the places that I would have liked to have gone, which I didn't, was to go to the United States to work with a chap called Marvin Carruthers. And Marvin Carruthers developed the chemistry which is used in oligonucleotide synthesizers. He'd worked with Gobin Khorana, and he just moved to Boulder, Colorado, and set up his own research there. But the chemistry of oligonucleotide synthesis that is used throughout the world to make any primer, he developed that chemistry. And so I wanted to go work with him. Unfortunately, he didn't have any money, and so there was no way potentially for me to do that. So I didn't go out to Boulder, Colorado. But then, probably about a week later, my boss said he'd been chatting to somebody at a meeting, and he would take me to do a postdoc in his group in Denmark. And this was with a guy called Brian Clark. Brian Clark had just moved to Aarhus in Denmark about 4 years before, and he'd been in Cambridge, and he was part of the group that did the work on the crystal structure of the transfer RNA molecule. And his group was then looking at the proteins that interact with tRNA, and this was one of them was elongation factor TU. So they were doing the crystal structure, and so he said I should, would I be interested in going out there? I met Brian at a meeting in London, and we had a brief chat, and he said, "Yeah, fine, yeah, so you can come and work for me."
Perfect.
And so I had to apply for an EMBO fellowship. I do feel bad. I did apply also for a Royal Society fellowship, and I was awarded it.
Wow.
But I turned it down.
Oh.
Because it was only for 1 year, whereas the EMBO fellowship I applied for was for 2 years. And so I knew I was going to have money for 2 years working with Brian. And so that was that. I had to go to Paris to be interviewed for my EMBO fellowship, which was, for me, was fantastic. Here I was, just a PhD student in my final year, being flown to Paris to be interviewed for getting this EMBO fellowship. And
so why Paris? That's an old location.
You had to be interviewed by a member of the EMBO organisation, the European Molecular Biology Organisation, and in those days it was probably much smaller. And the person in Paris was a woman called Marion Grunberg-Monago, and she was very friendly with Brian Clark, and she was the one that got to interview me. So I flew out for the day, had a little chat with Marianne about what I'd done and what I was going to do, and then in typical French fashion, we went out to spend 2 hours at a meal in a restaurant, and then we came back to her office and we sat down and I helped her fill in the form for my application, making sure she ticked all the right boxes, which was a bizarre situation to be in. And then I got that EMBO fellowship and went out to Denmark for 2 years.
So what was your experience in Denmark? And do you speak any Danish?
No, but I went to Danish lessons for 2 years. You quickly learn when you go to Denmark that the Danes speak English perfectly. Even the sort of the road cleaners, anybody, the shopkeepers, anybody, they all speak perfect English. So there was no need to speak Danish. And obviously working with Brian's lab, Brian came from Wales. So English was the language there. We would try and go into the lab and say, right, this morning we'll actually all speak Danish. And by coffee time at 10 o'clock, it just fallen apart, and they're all just speaking English. So that was quite an interesting sort of experience, going to another country where English wasn't the first language. But Denmark was fantastic. I learned so much there, had a fantastic time. And I went there with my wife, who didn't have a job at the time when we arrived there. I had an EMBO fellowship, and we arrived there, and got chatting, and Brian asked Claire, my wife, what she'd been doing, and she'd actually worked as a technician in Birmingham. And so he just rang up his friend in the Molecular Biology Institute and said, he's got somebody here who would be perfect to go work in his group. And so the chap said, yeah, fine, send her along, we'll have a chat. And she was offered a job there, so she worked there for 2 years nearly.
Oh, wow. So you were both there for 2 years?
For 2 years, yeah. And as I said, Brian's group was doing the crystal structure, but at the time they didn't actually have the amino acid sequence to fit to the crystal pattern, to the X-ray diffraction pattern. So it was decided that I would go and work in the Molecular Biology Institute and do the DNA, they do the protein sequence of elongation factor TU. And so that was in the same group that my wife had just started. And in typical Danish fashions, they assumed that the best person to teach me how to do protein sequencing would be my wife, who'd just been taught it. And so effectively, for 2 years, we worked next to each other.
Yeah, I suppose that goes one of two ways. It's a make or break thing, isn't it? You either enjoy each other's company so much that you spend every waking hour together, or you find that you don't. So
no, that was we're still married, and it was actually a fantastic experience, to be there doing work, at a good institution, and it was really good. So Claire was working on blood coagulation proteins. I was working on elongation factor TU, and it was just incredible. And because of Brian's connections with the MRC, and also the boss of the protein group, Staffan Magnusson, had connections with the MRC in Cambridge. We got amazing scientists coming through, who were visiting Europe, that would come through Aarhus. So, we had quite a few pre-Nobel Prize winners coming, giving lectures, and we would have, we would talk to them afterwards. That was sort of really nice.
Anybody? Who is the most famous one that sticks in your mind?
Well, we had Max Perutz come to visit, and I ended up at one point in the evening sitting in the back of a car with him while Staffan drove us to have dinner with one of their scientists, and and that was really fantastic to be sort of just chatting to somebody. It was quite famous. The other thing that happened when I was in Denmark, which I suppose did change what I ended up doing, was that we decided, as well as doing the protein sequence, a scientist in Aarhus had the gene for EFTU cloned, the bacterial gene. So we decided that perhaps I should go to the MRC in Cambridge, to Fred Sanger's lab, and learn how to do DNA sequencing.
Nice.
So I went there for 2 weeks, and that really was fantastic, to be at the MRC labs, learning how to do DNA sequencing. And just being there was phenomenal. I mean, they had the policy that, that the top floor of the MRC building was basically the canteen. And people would go there, and they would spend hours in there, because they would sit and talk to people in other groups, and you would, discuss ideas and come up with new theories and such like. And it was fantastic just to be in that, just to be sitting there and realising that the person coming to sit next to you was Max Perutz or Aaron Klug. So you have these Nobel Prize winners who were just quite happy to come and just chat with you. And it was so uplifting, and being present there at this fantastic institute. So the point was then that I learned how to do DNA sequencing, and I thought, yeah, this is what I want to do. And I suppose that's what I've more or less done, ever since, was to use DNA sequencing to analyse gene structure and help people understand, gene function.
Sure. So then you took this newly learned DNA sequencing knowledge, and you took it back to Aarhus. And from Aarhus, how long then did you spend in Aarhus before you ended up in the city that you're currently in, which to our listeners is London? So how did what was the
that was about it was only about 6 months after I'd finished in Cambridge. I went back. We finished off the protein sequencing. We didn't get too much out of the DNA sequencing because we didn't have enough time, because at that time it would have taken quite a while. But I brought the technology back to Aarhus. And then when I was in that sort of 6 months, it was a matter of deciding, what do I do next? And again, through contacts with Brian, I I met Beverly Griffin, who was working at the Imperial Cancer Research Fund, and she was working on the mouse polyomavirus, which is a cancer virus, a mouse cancer virus, and she was working on that, and she would be doing DNA cloning, and she was just starting to branch into working on Epstein-Barr virus, which causes glandular fever, and it's also the virus which causes Burkitt's lymphoma. So that was quite a sort of an interesting for me to move into sort of DNA technology for looking at viral genes and viral viruses, and their oncogenes. And but at the same time, I talked to a chap called John Walker at the MRC in Cambridge, and he said, why? Because I've been doing protein sequencing, and that's what he was doing. He said, why don't you come and do a postdoc with me? And I thought, well, okay, it would be nice to go to the MRC in Cambridge, but I wanted to move away from protein sequencing, so I didn't go. Which I always think, again, is one thing we think, should I have stayed? Because John Walker got the Nobel Prize in the late 90s. So again, you just think, whoops, I would have been there at the time that work would have been done, that setting for his Nobel Prize. But I don't regret any of the changes or any of the things that I did. I had a fantastic time moving to work with Beverly Griffin. Mm-hmm. So I moved there and started working on cloning and analysing Epstein-Barr virus genes.
Okay.
And one of the things that was interesting at that time, because it was an oncogene and it was an oncovirus, causes cancer, you couldn't clone EBV genes in the UK. The genetic modification guidelines said that's not an allowable experiment. It's too dangerous to do.
Oh, wow, okay. Right.
But you could do it in Sweden.
Right.
So Beverly.
So you flew out to Sweden.
Yeah. So Beverly's partner Thomas was just hadn't quite moved from his lab in Gothenburg to come and work in London with Beverly, and so I went out to his lab in February for 2 weeks to do some cloning of Epstein-Barr virus genes there, and that again was a fantastic. It was just snow everywhere, fantastic setting, and Thomas took me back to his flat to have reindeer steaks. So we went back to his flat, and he used to write for a wine magazine. So we had 3 bottles of wine to taste, and reindeer steaks. And it was fantastic. I had a thoroughly great time. And the thing about Thomas is that he got the Nobel Prize for DNA repair recently. So
really, what year do you know?
I think it's about 4 or 5 years ago. Because he's the one that he pioneered the fact that DNA repair mechanisms, that DNA is not static. It gets damaged, and there is a repair mechanism to make sure that the integrity of the DNA sequence is maintained. That's one of the fields that he worked on when he came to London.
Oh,
but I worked with him when he was doing some simple EBV stuff. So I came back.
That's incredible. So that's 2 Nobel Prize winners who you've, or future Nobel Prize winners who you've associated with, and then.
Yeah, and I say there wasn't anything, that's what happened. Lots of people would in those days come across, Nobel Prize winners, and, you chat with them, because the field was much smaller. It was a much smaller, I think, biomedical, molecular biology field. So I then worked with Beverly cloning and analysing Epstein-Barr virus genes. Again, that fellowship was just for 3 years. I then moved to do an MRC postdoc in the chemistry department at Imperial College at South Kensington, where I worked on site-directed mutagenesis, and the group I was working in was fantastic.
Did he go and get the Nobel Prize, or she go and get the Nobel Prize as well?
Well, I wouldn't be surprised in the future if Alan didn't. But effectively, what they were doing, we had the crystal structure of a protein which binds to tRNA. And we had the gene and being cloned, and a group in Cambridge were making mutants based on the crystal structure. And what Alan's group was doing the kinetics. And this was the first time that there was a logical link up between, we have the structure of this protein, we know what the protein does, we can make modifications to the protein, and then we can see how do they actually work biologically.
Okay.
If you change this amino acid in the protein, how does it affect the activity of that protein? And so he was doing the kinetics. So between the three groups, the crystal structure, the making the mutants, and doing the kinetics, you can actually get an understanding of how that protein was really working. And it was one of the sort of the very early days of doing side rotating mutagenesis to understand protein function.
Yeah, it's crazy. So didn't quite recently Frances Arnold from Caltech, she won the Nobel Prize for speeding up the directed evolution of enzymes. Is that a similar process with the site-directed mutagenesis?
Well, it is. With the site-directed mutagenesis was that you had the structure and you would say, okay, the protein does this function. What would happen if we changed that amino acid? So knowing the crystal structure, you knew the binding site for the ATP, which was used as the energy for driving the reaction the enzyme was involved in. So you could say, well, actually, if we change this amino acid, how does that affect the binding of ATP to the protein? Does that make it bind better or worse? Does that affect the kinetics? And that's what scientific mutagenesis is doing. You're trying to make a variant and see, does that make it better? Does that help you understand how that protein works?
So what protein which protein was it that you were working on?
It was a transfer RNA. So basically what the enzyme did, it bound to a tRNA, and it bound the amino acid, and it attached the amino acid to the end of the tRNA, so the tRNA can bind to ribosomes for protein synthesis. So it was it was binding ATP, a tRNA molecule, and a specific amino acid to attach to that tRNA. So you wanted to know, how does that protein recognize a tRNA, because it's only going to bind to a specific subset of tRNAs to put that amino acid on. So it was all understanding that. But the concept was there that you had a crystal structure, you could make variant enzymes, you could then do the kinetic analysis to understand what is the function of that protein. And it's basically what you should be doing with every protein, to understand how those proteins work. But it was very early days. And it was interesting because you had a kinetic assay, so you could understand how changes subtly could have changed the activity and the efficiency of a protein.
So then, I'd like to close this section just by asking what your advice would be to young people at different stages in their career. So throughout all of the experience that you've picked up, what would you say to a young person who's about to enter? I mean, in the current climate, firstly an undergraduate degree, then what do you think about postgraduate degrees, and then post PhD positions?
I think my advice is more or less the same for all of them. Do something which you enjoy.
Yeah.
I mean, one of the things I've done in recent years is has been on the interview panel for students coming to Imperial College to apply to do medicine. And with a lot of them, you can pick up on the fact that their parents have decided you should do medicine. We want a doctor in the family, and you feel that some of the students have been they're being directed into a field that they may not necessarily have wanted to do themselves. And I think you've got to do something that you feel really interested in, that you potentially become passionate about, and you enjoy. I think it's pointless trying to pursue any degree, whether it's BSc or a PhD or even beyond that, if you're not really interested and excited about the field. And for me, it's been so nice that I've had that opportunity to do what I've been interested in, and also been paid to do it, which I find is quite amazing. But I think the advice is actually do something that you really are interested in, because I don't think because you choose a particular undergraduate degree, that does not necessarily mean that you're now committed to down a particular pathway and you can't change.
Yeah.
Because I did basically oligonucleotide chemistry for my PhD, But then I changed completely for my first postdoc to do protein chemistry, protein sequencing. And then I changed for my next postdoc to do DNA cloning and DNA sequencing. And I found that it wasn't difficult to change, because I felt that because of my enthusiasm for learning and the fact that I must have a certain amount of good ability in the lab, I could apply myself to whatever new field I wanted to move into.
I think that's actually a crucial a bit of advice for people who are a little bit later on with their studies, like postgraduate or thinking about their first post-PhD position. It's that, you're only going to get one chance at being, a freshly graduated PhD student. So what I feel is that if it's really good advice for a postgraduate student who is looking to go into something a different field, because the idea would be that you don't just learn the same things that you did in your PhD. You try and learn something a little bit new or a little bit different, because that's the only way you're going to ever expand your horizons. You can become an expert in one tiny little field, which is great, but at the same time you have to expose yourself to new environments, because if you don't, then you're not going to learn what else is out there, and you could be better suited to something else.
Yeah, it's I guess it's that, I suppose, that first step, that first BSc degree, what do you do, is and I did it because I was good at chemistry and I like chemistry. That was sort of it. It was something which I enjoyed doing.
I think that's the best reason. I truly believe that's the best reason. And like you said about, you don't have to go into further study. You don't have to do a PhD. You might have familial pressure, but you don't have to. You always have a choice. You've always got a choice. So
I feel doing something which you enjoy is important to do that. Because it's a lot of work, and you put a lot of effort into it. For your degree, PhD, and postdocs. So you've got to be able to. You've got to feel that you're going to enjoy it, and I can probably think back since I started my PhD until today that there's probably only been about 10 days where I've not wanted to go to work. Where I thought, God, have I got to go to work today? Otherwise, for me, I've been excited, I've enjoyed the fact that I'm going to come in and meet people and go into the lab and do that. It's been a pleasure. It's not something which I've dreaded. It's not like I think, I've got to do the same thing again today that I did yesterday. I've gone in thinking, well, okay, let's go for it.
Well, that's, with being retired now, 10 years and an entire career of not wanting to come into work that's not bad going. That's not bad going at all.
Yeah.
For the next section, I wanted to discuss your experiences in science. So, when you were a student, what was your most memorable experience?
I guess the undergraduate, I think it was just the fact that I think it was the whole thing that every afternoon during term time, so Mondays, Tuesdays, Thursdays, Fridays, 4 afternoons a week, you were in the lab doing experiments. And I guess for me, doing organic chemistry was fantastic. Being able to, in the lab, that was the fun thing, doing experiments, actually making chemicals. Okay, you're making the smells and such like that you associate with some chemistry labs. But that, to me, was the thing which made it really interesting, being able to do that. And I guess it was the organic chemistry labs that were the most things which I found really exciting to do.
And then after you graduated as a student and throughout your career, what has been the highlight, or what's been the most memorable thing? 1 and 2 might be the same thing.
Yeah, I guess some of the work we did for doing the protein sequencing as a postdoc was really exciting, in that we could fit the amino acid sequence into the crystal structure, see what the protein looked like. That was really a nice piece of work that was finished and was done beautifully. Similarly with some of the cloning experiments I did as a postdoc, it was fantastic to sort of to finish that when we've helped finish the sequencing of Epstein-Barr virus and human herpes virus 6. When you finish the sequence of that and you got the final genome sequence, that was fantastic. That we helped to do that. We didn't do all of it, but we did our part, which was together with the other scientists that were working on it. I guess as for a PhD, it was interesting that we did try some interesting stuff. My PhD, the idea behind my PhD project was to develop interferon inducers. So you've got to think back. This is the early 70s, where interferon had just been discovered, and interferon was something which actually would, if you induce that inside a human body, it would help fight virus infections.
Okay.
And it was known that if you took a short, a sort of a artificial double helix of DNA or RNA, which had inosine in one strand and the base paired with C in the other strand, if you just took that homopolymer and you added that to cells, it would induce interferon. Mm-hmm. And the problem with natural nucleic acids, they're negatively charged, they're not taken up very well by cells. So the idea of my professor was to make an analogue of a nucleic acid where you didn't have a phosphate link, you had a neutral peptide link. So you'd have a double helix, but it would be neutral, and hopefully would be taken up by cells more readily, and therefore potentially would induce the interferon response better. So that was our simple concept.
Just a curiosity thing then. How did you actually, what was the delivery factor? Did you have a...
No, that was just basically just taking chemicals to make basically try to permeabilize membranes. Then you just added your oligonucleotide in solution. It was such a simple thing. You took your naked DNA, added it to the tissue culture, or injected it, or just injected it into animals.
So there's no nanoparticles, there's no liposomes.
No. It was just stick it in a syringe, in a needle into, and so I spent my 3 years trying to chemically make a poly C analog. I eventually made some, and then in my last couple of months, I actually went to GD Searle, which is a biotech company in High Wycombe. So I went down there with my oligonucleotide to test it out on animals. And we basically gave mice a lethal dose of EMC virus. And I'd never done any animal works at all. So for me, it was exciting to go around and see animal experimentation. And this virus, when it infects mice, it causes paralysis, and the mice usually die. Mm-hmm. So I went down with my chemical, and they set the experiment up. So many mice had the virus, so many mice had my compound, so many mice had my compound and the virus, and all the various controls there. And we did that injection, and then a couple of weeks later I got the results, and it turned out that all the mice, if a mouse had the EMC virus, then 70% would die, 30% would survive. But with my compound, all the mice died because it was lethal.
Oh no!
So it was a slight disappointment that our analogue actually was poisonous. But still, that's science for you. You don't necessarily always know what you're doing.
It is. But people have to remember as well that you don't know what you're doing. It is research. You do something because you think it's the best thing to do at the time, and it's obviously that if you look back in hindsight, it's a wonderful thing, hindsight, because I should have done this and this. You knew that would obviously not work, but you have to try it, and it must have been really exciting that such faith was placed in your work that could go forward. But then, like you mentioned, maybe a little disappointment that it didn't.
Yeah, it's. I mean, say some of the work actually was quite useful, because I think some of the analogues that other chemical groups worked on, putting phosphoramidate links in to oligonucleotides to make them more stable, have been used for making oligonucleotides, which are tried to use for biological treatments. So so the chemistry that we developed, there was a use for it in other things. Not the one that we were particularly working on, but the general field of doing nucleic acid chemistry, making nucleic acid analogues has been useful.
So what was the biggest challenge that you faced as a student?
I think the biggest challenge is realising that not everything you do is going to work. And that you've got to just do something, it doesn't work, you've got to think about it and then just try it again and keep trying. And not basically be put off by the fact that it's not working. You just got to keep trying and work out why it's not working. And I think that's the biggest challenge, is that if it doesn't work, why hasn't it worked? Is it because I'm so stupid I put 5 microliters in when I should have put 50? Or is it because actually our theory is not quite right? We need to rethink about what we're doing and why we're doing it. But I think you've got to experience the fact that not all your experiments are going to work. And I've told many students over the years that I suspect that if you looked at all the experiments that you did in a year, if they worked first time, all those experiments would have probably just taken you 2 months.
Yes,
and it's understanding that that's what you've got to be doing. You've got to be working at it and doing it time and time again till you get it right, to sort out what the problems are. It's not going to be perfect. And in fact, I would feel that as a scientist, you need to have those struggles. Because if it worked first time, every time, then I think it would be a bit boring.
That's true.
The science.
You need
to struggle. You need to basically realise that it's not quite right and work it out there. And I think that's the best advice I would give, is that that's what you're going to come across, and you've got to work at it to actually overcome that to become a good scientist.
True, very true. But then, what was the best piece of advice that somebody ever gave to you?
I think the best piece of advice probably came from Beverly Griffin, who I worked for twice. And that was basically that when you're sort of recruiting people and working in a group, you've got to work in a group where you all get on. And that you've got to choose people that you like, that you become friends with, and that you enjoy working with. It's because that makes the science interesting and exciting, that you've got people that you can share your failures and your successes with. So I think that is actually making sure that you actually work with people that are like-minded, that you get on with. I think that's the best advice. We've had situations where we could have employed somebody who was on paper looked incredibly bright. They're going to be the next Nobel Prize winner. But when you actually talk to them, you realise he's not going to be a nice person to work with. He's not going to fit in. And I think that's the thing, is to make sure you fit into a nice group. And we would sooner have a group where we all got on well, because that way you're going to be more productive, than actually if you didn't get on with people in your group. You were always falling out and you didn't like them. That's not good for science. It's not good for people's careers. I think that applies to everything, I think.
It's also not good for your stress levels as well. Like,
yes.
you want to, you might be able to produce this cutting edge science, but if your life's miserable, then what are you doing? why are you bothering? Yeah.
That's what I'm saying. That's why I said I've always been pleased that I've enjoyed everything, and that's what's made it exciting. And it's not just the fact that I've done good work, but I've done good work with people that I've enjoyed working with. And I think that makes you better and makes the work more interesting and also more successful.
Very true. Okay, so this is a tough one. It depends on how honest you want to be about this one. But what was the hardest decision of your career?
It's difficult. I mean, as you probably would realise, that each time I was making a move, it was all more or less word of mouth, in the sense that my bosses would ring up, they would phone up somebody and say, "Look, I've got this great guy working for me. He can come and work for you. He'll be perfect in your group. There, can we fix it up?" is this okay? Go meet and talk with him. So there's never been me really applying for something and not getting what I've ended up doing. Whether I'd done something different, I don't know. I would like to think that even if I'd say gone to Cambridge instead of going to the ICRF, then that may have been. I would probably wouldn't be at Imperial now, but I think I would hopefully would have enjoyed my scientific life just as well as what I have done.
Yeah.
So I suppose that probably was one of the sort of a tough decision to say, well, am I going to go and work in London in a new area doing gene cloning and gene sequencing, or go to the MRC LMB Cambridge with its fantastic track record, and work with somebody who would eventually become a Nobel Prize winner. And that, I suppose, was a tough decision to make.
It's very tough.
I made it purely on the basis that I wanted to do something new. I wanted to be in the DNA field. I thought that was where the next big breakthroughs, the big interesting science was going to be. I would actually, in thinking now on hindsight, I think actually the one field area, I think doing protein sequencing is still now more important than, as well as sort of the gene sequencing that's been done. Because we can analyse genes now trivially. It's really quite easy to find genes and sequence them. But actually working out the structure of the proteins, that's the key thing for understanding how it all works.
Yeah, so a lot of people are taking multi-omics approaches. So just to explain what that kind of means, is that you would have proteomics, which is sequencing the whole proteome, all of the proteins that are involved. You would have transcriptomics, sequencing all of the RNA transcripts. You would then have things like metabolomics. So all of these are different processes, and they're all intertwined. So it's very, very relevant. This multi-omics approach is that's still pretty cutting edge today. So it's not that this is a bygone technology. This is still very, 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 very relevant.
Yeah, but I think at the time, in I guess the 80s into the 90s, doing the protein work sort of fell by the wayside, and the core enthusiasts thankfully maintained working on that, because everybody thought working on DNA will be the way that we will solve things, and I think they realised it can, but you still need that sort of proteomics, that side of it as well, and it's not just one field will do that, sort of one area.
Yeah. So okay. Final question in this section is about your top work-life balance. So a lot of people have a lot of issues in terms of balancing work because of the need to work hard, the need to prove yourself, the need to sort your PhD out, and or your bachelor's degree. And a lot of people put a lot of hours in, which is why it's such a great achievement. But work-life balance is one of those things that is sometimes forgotten about. What would your advice be to people to try and help them maintain one, or to strike one just in the first place?
Well, if we go back to when I was doing my degree, I started going out with my wife at the time.
This is your bachelor's degree?
My bachelor's degree, yeah. She was the year below me, and we started going out. And then I studied my PhD, and so Claire remained in Birmingham and it was a technician's job, so we were together. We got married while I was doing my PhD, and actually, because in those days, it was expected that PhD students would be at university during term time, I had to get sort of permission to actually not be at term time when I was getting married. So I usually joke that I had to get permission from my head of department to get married. So I got married when I was doing my PhD, and then obviously we went out as a postdoc. And I think having somebody close that you can share the ups and downs with, I think that's so, for me, was so important, that there was somebody that you could go home and say, bloody hell, that experiment didn't work again today.
Yeah.
And I'm sorry, I'm not coming. I've got. I think having somebody there that's supporting you, and you're supporting them. I think that's so important, that you're not on your own. That you're not feeling that I haven't got anybody who I can basically cry on as a shoulder.
Yeah.
And I think that to me was so important, so useful. The fact that there was two of us. So I feel like although it was my PhD, it was also Claire's in a way as well, because she's helping me mentally, get through all the hassles that we actually, you come across, which, you on your own, I think would have been so hard to overcome. So to me, that's the best thing, is having somebody there that you're sharing all the successes and also, the downsides as well.
Definitely. How old were you when you got married during your PhD, just out of curiosity?
I was 24.
24. Okay, fair enough. Okay.
Yes. Yes.
No, it just must have been like to get married during a PhD. It's a nice break from your work, right?
Well, again, I mean, not that but again, I was thinking that in the group that I did our PhD in, I think probably 4 of us got married while we were doing our PhD, or were married, there. So it was. I won't say it's what you did when you were doing your PhD in Birmingham that you got married, but it wasn't an unusual thing for you and your partner to be married and doing that. So that was the thing, and you do, and also at the time, I know of so many people that it was husband and wife team that were doing the research as such. So.
Yeah, so having a supportive partner, whether or not they work in science or not, I suppose is really crucial.
Well, I don't think it matters. I think it's probably having somebody who works in science, they can probably understand,
the jargon and all of that.
The jargon and such like. But I think it's more or less somebody that you feel that you can switch off, going home and being able to just switch off and do something. Go out to the pub, go out to the cinema or whatever. I think it's having that. I think is important. And I mean, I know from talking to colleagues that went and did postdocs in the States, is that I've always had the ethos that you do an experiment. If it doesn't work, you basically say that's it. You go home and you think about it. And then come back the next morning and discuss it with your supervisor or whatever, and then start again. Whereas I've talked with people that went into the States, and then the American attitude was that, your experiment doesn't work at 5 p.m. in the evening. Well, so what? You just start again and work through the night. And I think that sort of work, that you work all the hours and you just keep on working, I think that sort of attitude is not helpful all the time. I think you need to have a situation whereby if something doesn't work, you basically stop, take stock, think about it, take some time out, not just basically say, okay, I'll continue, I'll start again now.
Yeah, definitely. What are your passions outside of science? What do you like to do in your leisure time?
God, up until now, leisure time was doing science. It's always been for me is that because it's sort of a career. It's in a sense you're never not quite really switching off. It's not like a job where basically when you leave work, that's it. You can't do anything. When you come, as a scientist, you can always think about things. You can always sort of plan, write papers, write your thesis. You can always do things. So I think for me, I've not thought, I'm going to now switch off from work and do a hobby.
Yeah.
But what I really like actually is visiting places. London is fantastic. There's so many attractions. From the museums, just going around the markets. Going around Borough Market, choosing a nice cheese on Saturday mornings or things like that. It's just actually getting out and experiencing London life. I think is one thing that I've thoroughly enjoyed because I think London is quite a unique city to be in,
Yeah. What's that famous quote? It's something like, "When a man is tired of London, he's tired of life."
Yeah, and I think
Samuel Adams. Yeah.
Yeah, I mean, so that is such, I find that such a nice thing to be able to just do, to just say, right, okay, we'll take off. We'll obviously not now with the COVID restrictions, but be able to just sort of go and just enjoy going around London, even just walking around, Trafalgar Square or up through Chinatown. It's just really fantastic and interesting just to sort of enjoy what's on offer there.
Definitely.
Obviously, it's now that I'm thinking of retiring, I've got to try and perhaps choose some hobby, but I haven't quite got one that I would say, oh, this is my passion that I'm going to be doing.
Well, this is what I was just wondering. Then, do you do different kinds of science in your hobby time? Do you make a plasticine volcano, put some baking soda in, and stick some vinegar in?
One of the things which we have been doing recently, again because of lockdown, is doing jigsaw puzzles.
Okay.
So I've just finished I think it's about 1,000. I don't know how many 5,000 piece jigsaw puzzle of basically it's like a Hayes manual for a Spitfire. So basically, you've got the Spitfire with all the intricate bits that go inside it. The only snag was probably 300 pieces were just all white. With nothing on them. But it's interesting things. The other things I'm starting I will probably do as an interest is genealogy, to go back and find out what my parents and grandparents and great-grandparents, what they did. Try and go back as far as I can.
Yeah, like a "who do you think you are" type
thing.
thing. Yeah. So I also had it sort of got put on hold, but a few years ago I had my own sort of like drive to want to do something like that and go back and trace my family tree and figure out who did what professions, like, because I have some family that emigrated to Australia at some point. I've got some in Canada, and I know like different surnames, but it'd just be interesting to be like, okay, like, I'm related to you. What do you do? I don't know. It just got put on hold because of my PhD. So. Retirement.
That's right. I mean, I did dabble a while ago, and the complication with going down my father's side of the family, that his father, Samuel Jones, married a woman called Elizabeth Jones, which makes trying to go back through the history, the genealogy, and you sort of the censuses when you're just Jones is quite tricky. I have more success going down my mother's side, because she was a Fox, so it's the case in that FOX, the surname. That's been it. But that's something that I think I will probably sort of take up. But again, it's not something. I just feel that it's just getting on with life and enjoying things.
That's true. So, are you a passionate advocate for anything?
I
mean, you don't have to be, by the way. It's just
I think probably it's probably trying to get science understood by the general public, to understand that there's some science that that is so useful to have that knowledge, particularly I think in the sort of biomedical field. I think you've only got to try and explain to people about the COVID vaccines. And I think that's something which, being able to get that across to the general public will be something I think is really important, because lots of people have got totally the wrong idea about about vaccines and what it can do and such like. And I think, getting people to really understand and appreciate how science has helped, how we are where we are today and in health, like, is so important. So I think that's something that I would be, would want to push. But obviously, there's thousands of people out there already, sort of doing that as well.
Yeah, well, you're doing your bit here. You're doing your bit by explaining to all the next generation of young scientists how the trials and tribulations of everything. So it's definitely important. And I also find that communicating science is quite difficult sometimes, because there can be an unwillingness to learn, but there can also be a resistance to even be confronted with something that doesn't fit a particular narrative that is being fed in mainstream media, and with all of this, it gets really tough. So I think there's a lot of disinformation and things working against the grain.
Yeah, it's also interesting. I find that when I've watched, say, Horizon programs, if they've been about subject matter, particularly say like physics, astrophysics, things which I know nothing about, I get really interested and I can see the excitement of it. When you start to have a Horizon program that's on something that you know a lot about, you start to realise, well, that's been a bit simplistic.
Yeah.
Yes, and I think that's the problem with scientists, that we know too much, and trying to get the ideas across to the public in something that they will understand is quite difficult to get them. I mean, I quite like to tell to get people to try and appreciate sort of size in biology. So one of the things that I've told our non-scientific friends is that they can appreciate how small a single cell is. It's very small; they can't see it. But I tell them that if they took the DNA from that cell and laid it end to end, that length would be 1.9 yards, 1.9 meters from a single cell. If you laid your DNA end to end, you can do the calculation. You can work out the distance between a base pair and work out the number of base pairs there is in an average cell, and that's about just under 2 meters.
That's crazy.
And
all of the cells in your body, then, how far does the DNA stretch?
Well, I think somebody, actually one of the lecturers at Imperial does that in one of his slides. I think it's actually to the moon and back. It's ridiculous. And then the other one that I normally tell them about is that people appreciate that that they've got bacteria. They've got healthy, good bacteria on them. You're living in gut bacteria, and all around their body, in their noses and what have you. And I tell them, if you counted up the number of human cells in your body, and then you counted up the number of bacterial cells in your body, what would those numbers be like? Do you know the answer?
Yeah, so I do, annoyingly. It's that, there's more bacteria in and on you than there are cells of you. So you are more bacteria.
Exactly.
Yes.
And again, it's actually getting them to understand these, I think, will get them interested in science. That science, it actually is quite exciting and interesting. But you've got to be able to I think, to get it, to tell it in such a way that they can understand it. Because they're not all, most people will have forgotten all their sort of biology and chemistry if they're not doing it. Everybody that's sort of left with O levels and A levels, they will have, by the time they're in their sort of 50s, they've forgotten about all that sort of stuff. And to try and get them to understand it and to show that how it's important and useful.
Yeah, but I think that's what
you've got to get across them.
But first, you need to just re-engage. So I think understanding comes maybe second or third in the line. You need to just re-engage them and get them interested, because I think understanding comes from interest. Like with all of your career's work. You first heard about something, you maybe had an idea about what the concept was, but it was only when you got truly interested in it that you truly started to understand it. So I think it's generating that interest and the enthusiasm, which that's pretty tough. I don't know how to do that one. If there's anyone out there who does, get in touch.
Well, I would say you do see with some of the scientists that are on the television that sometimes they get it really right, and you think you can see they can be enthusiastic. David Attenborough, spot on. You want to get him to tell everything that we want to get him to add.
Oh gosh, it's going to be a sad day, isn't it, when he's no longer with us? He's complete gem. Okay, on to your final thoughts. So it's been a very challenging 12 months almost now, and everybody is struggling. It's plain to see. What would your advice be to young people right now?
For that, I would say stick with it. There will be an end in sight. It is unfortunate, but and I think it brings home to people that with viruses, we can't control them. Well, we can with vaccines, but we would never have predicted. Well, I think maybe as scientists we could have predicted there would be viruses crossing the species barriers, and we may have a situation like this. But I think people, the general public, young people probably wouldn't think, why is this happening? Why is there suddenly this threat to our lives, which we have no idea about? But I still think the advice is that just stick with all the good advice that's coming out, and there will be an end in sight to this. Yeah, it's going to. I think it's going to take a while, and I think hopefully get them to appreciate that all the science that's making the vaccines possible is phenomenal. I mean, if this virus crossed the species barrier 30 years ago,
no, we don't know. We
would be in a much worse situation. We probably wouldn't have even at this stage maybe got some of the first bits of sequence from it. We might not be getting the vaccines, would not be being developed. It's all the technology. So I think the advice would be trust the technology. Trust the science. It's going to work for us.
How do you see the future of your field?
Well, I think the future is, in a sense, is here now. The ability to analyse genomes. We're seeing phenomenally with being able to sequence more nearly instantly new COVID strains, and I think that is impacting on how we have new vaccines and new treatments. So I think that is really, got things really exciting and interesting in that technology is there to make short work of tackling some of these biological problems.
So, but I kind of want to get an idea of what you think, would be sort of the technology, not that's completely futuristic, but perhaps maybe beyond your years, in terms of that if you were to think 30 years or 50 years ahead, we've got all of this sequencing technology, like, let's say we mission completed. We have finished sequencing. We have done it. We know everything. We know the sequence of everything, of all life on Earth. What's next?
I'd say actually, if you look on from a medical sort of health perspective, it's effectively personalised medicines.
Personalised, okay.
But it's beginning to be here, but I think that's going to be the future. Where at the moment you have a headache, you go to the GP and he says, "Okay, you've got a headache. I'm going to give you drug A." Because that's what we give people with headaches. But what you'll find is that in the future, if you know your DNA sequence, it may very well be your genetics mean that you being given drug A, it's not the best drug for you. You need to have drug B or drug C. And you don't prescribe drug C because only a few people really need to have drug C. Drug A is better. But if you knew your genetics and how your genetics impacted on how well a drug worked, you go to the GP with your headache and he'd say, yes, okay, you've got a headache. Okay, I can see your genetics. You need to have drug C, not drug A, because they would give A first because that's the one that most people would take. So you'd actually have a situation whereby you'd go there and depending on what your genetics are, you'd have a particular drug treatment which would be different for everybody else with the same condition because your genetics will impact how well those drugs work in your body.
So yeah, to be honest, I totally agree. But then, so you've got minor ailments being cured, shall we say? Will we be living longer? Will we be having, for instance, the whole telomerase shortening is apparently associated with aging. Do we live forever? Or?
I suspect probably not. But I suspect we probably will live longer. I mean, it's quite clear when you look at, and again, that's probably down to health in terms of getting rid of bacterial infections and some of the diseases which can now be cured. Hopefully, sort of cancer, and other diseases. These things are going to be like dementia, old age diseases that are now going to be the problems. And how can they be treated and handled? And I do wonder whether, in actual fact, life as we know it, will not necessarily be able to live to be 200.
Yeah.
Maybe 100, 110, maybe. It's difficult to understand how you would do that, because then you should imagine that if you did live to be 200, then think how many great grandchildren you got to buy presents for.
Yeah, I mean, overpopulation as well. I mean, I didn't think about this, but yeah, Elon might have colonized Mars by then, so maybe we'll be a multi-planetary species.
I think I think one's got to also appreciate that in the future you've got obviously the environment, but overpopulation is going to be something which is potentially the issue when we actually we can cure all ailments. When people are not dying from disease, then the population will be dying off.
It'll explode, won't it? The population numbers, yeah. But okay, we've sort of covered it there, but I just wondered if you have any emerging trends in science in general that you feel have limitless or unbelievable potential. I guess this goes past potentially what you are working on. I think actually the fact that a lot of science and a lot of research deals with things in 2 dimensions. Mm-hmm. When you're culturing cells and doing things, they tend to be in tissues, in sort of petri dishes, and it's quite difficult to do experiments and research on 3D structures. But I think that is what's going to be the most exciting future in sort of, I think, biology and medicine, is that we won't just be working with a single strain in a flat petri dish. We'll actually be working with an organ, and we'll be able to do the experiments we do on cells in a tissue dish. We'll be able to do them in a 3D organ and analyse what's going on and understand what's going on. I think that, to me, is the real breakthrough, is to be able to understand what's going on in a cell and how that cell interacts with other cells in the body and how the whole organ works at the sort of molecular level. I mean, the one of the issues I do actually have, and you come across it in looking at DNA sequencing. Mm-hmm. I think as an individual person, I can get my head round the 500 or so genes in a big virus like Epstein-Barr virus and herpes viruses and such like. And even with bacteria, you can get your head around where you can understand how a lot of those proteins interact. Mm-hmm. But try to understand that for a human animal, how everything interacts. I think by the time you work on a new bit, you've forgotten what you learned there, because I don't think in your head you can get your head around everything at once, which is needed. And that's the difficulty, is that you'll only be actually being able to understand and be effectively an expert in a small portion of the biology of an animal, not really understand everything in the detail that you understand, say, liver and liver diseases, to be able to understand the whole body at the same level, and you to understand it is quite, I think, is difficult to get, for me to get my head around. Would anybody have the brain capacity to understand it? So I think obviously artificial intelligence. Yes, I was just about to mention.
Comes into that, so we will be replaced by robots probably.
Yeah, I think it. Well, I mean, I hope at least. I'm not an AI expert, but I hope it's just an assistive tool, and that they don't take over, and Terminator comes and all of that. But but yeah, and obviously, in terms of, I mean, I'm going to be biased. I love the whole 3D cell culture aspects and the potential that's got. So I'll just leave it at that for now. But I definitely think that the AI the fusion of computer science with all of the traditional natural sciences. That is that's good. That's going to be truly crazy. All
Definitely.
Right. Definitely.
Okay.
So, very, very final question. If you could do it all over again, what would you keep the same and what would you change?
That's a tricky one. I'd be tempted to say I would do this more or less the same because I've enjoyed it.
Yeah. You wouldn't go back
enjoyed it.
You wouldn't go back and go and work on the Nobel Nobel Prize project?
Possibly, but the thing is that it's a question. It's the tricky thing about doing time travel that you can't necessarily go back, and I would feel that even if I had done that, then if I was being interviewed, having done work for the MRC Cambridge and not gone to the ICRF, then if I was having this interview again, I probably would still be saying I would do the same as what I've done to get me to that point. I think I've had fantastic opportunities. The ones I've chosen have been great. I thoroughly enjoyed them. Therefore, in a sense, I can't see what I would do different. But I don't think I would be agonising, oh my God, why didn't I do that?
Yeah.
I think life's too short. It's actually get on and enjoy it and do what you really think is fun.
I think that's a perfect note to end with. So thank you very, very much for giving up your time to speak to us today. Dr. Jones, thank you.
Not at all. Thanks a lot. You take care.
Well, there is so much to digest from this episode. Such an incredible life and career. I'm really grateful to Mick for sharing his experiences and for giving his advice. Please make sure that you're following us on Twitter and keep subscribing to our YouTube as well. So if you could please rate us on your podcast platform, that would help us out a huge deal with the metrics, and it'd be very, very much appreciated. Making a Scientist is going to start exploring alternative career paths, and I'm going to be joined by Dr. Vicky Sherwood, aka Biomed Badass, where we're going to talk all about what it means to be a scientific writer, and we'll discuss all about Vicky's transition from a PI to working in industry. Until then,